Tonight's lecture is over Chapter 8, Chromosomes and Cell Division. And our learning objectives for this lecture is to be able to distinguish the different types of cell division, to discuss how, through mitosis, worn-out old cells are replaced with fresh new duplicates. We will also be able to explain how sperm and eggs are generated through meiosis. We will be able to describe the sex differences in the chromosomes, and we will be able to discuss the consequences of deviations from the normal chromosome number. Throughout most of your body, your cells are continually dying off, and the ones that remain divide and replace the cells you've lost in an ongoing process. But can this cell replacement go on forever? And does a cell even know how old it is? Moving on. Actually, a cell does have a feature that provides an approximate measure of how old it is. Just as a car comes with an odometer which keeps track of how far the car has been driven, animal cells have a sort of counter that reflects how many times the cell has divided. This counter is a section of non-coding repetitive DNA called a telomere. The telomere serves as a protective cap and is located at each tip of every chromosome, right next to the genes that direct the processes that maintain the organism. Every time a cell divides, making an exact copy of itself, its DNA divides as well. However, each time the DNA divides, the process by which the chromosomes are duplicated causes the telomere at each end of every chromosome to get a bit shorter. At birth, the telomeres in most human cells are long enough to support 80 to 90 cell divisions. In a 70-year-old person, the telomeres are only long enough for about 20 to 30 divisions. Eventually, the telomeres can become so short that additional cell divisions cause the loss of functional essential DNA, and that means almost certain death for the cell. Occasionally, individuals are born with telomeres that impair the functioning of a protein that helps maintain the nucleus of cells. This genetic condition, called Hutchinson-Gilford progeria syndrome, causes the cell's telomeres to be much shorter than normal at birth. In addition, the normal functioning of many genes is disrupted. Consequently, cells and tissues begin to appear aged very soon after birth. Children with this disorder rarely live beyond the age of 13. Given this information, you might wonder if, by rebuilding the telomere after each round of division, the cell and its descendants could function for a longer than normal time. Such a line of cells would never die. By extension, it's tempting to imagine that constantly rebuilding telomeres might act like a fountain of youth. Unfortunately, it does not. Some cells do rebuild their telomeres after each cell division, restoring the chromosome's protective caps. For single-celled eukaryotic organisms and for the cells that divide to produce sperm and eggs in multicellular organism, that is, cells that must go through many thousands of rounds of cell division, this telomere rebuilding is essential. But for most of the other types of cells that rebuild their telomeres with each cell division, the telomere rebuilding presents a big problem. The cells are unable to stop dividing. Such immortal cells go by another name, cancer. Therefore, cancer is a disease of the cell cycle. And that brings us to our take-home message for section 8.1. Normal cell division is necessary to maintain most organisms. In eukaryotic cells, the telomere keeps track of cell divisions by shortening every time the cell divides. If telomeres become too short, additional cell divisions cause the loss of essential DNA and cell death. Moving on, all life on Earth uses DNA to store genetic information. This fact is remarkable considering the tremendous diversity of life on our planet. Ranging from single-celled bacteria to multicellular plants and animals, one way in which the DNA of different species varies is in how it is organized into chromosomes. Chromosomes are linear or circular strands of DNA comprising specific sequences of base pairs. The most important part of a eukaryotic chromosome may be the DNA molecule, which carries information about how to accomplish the processes needed to support the life of the organism. But eukaryotic chromosomes and some prokaryotic chromosomes are made of more than just DNA. The eukaryotic chromosome is composed of chromatids, a linear DNA strand bound and wrapped tightly around proteins called histones. This arrangement keeps the DNA from getting tangled and enables it to be tightly and efficiently packed inside the nucleus. Plants and animals usually have between 10 and 50 chromosomes, although there are species with as few as 2 chromosomes and others with more than 100. Moving on, most prokaryotes, the bacteria and archaea, have less DNA than eukaryotes do. They carry their genetic information in a single circular chromosome, a closed loop of double-stranded DNA that is attached at one site to the cell membrane. And when it is time for them to reproduce, they use a method called binary fission, which means division in two. This process begins with replication, the method by which a cell creates an exact duplicate of each chromosome. Replication in prokaryotes begins as the double-stranded DNA molecule unwinds from its coiled-up configuration. Once the strands are uncoiled, they split apart like a zipper with bases exposed on each of the two separated single-stranded circular molecules of DNA. As the double-stranded molecule unzips, enzymes bind to the DNA and attach free-floating nucleotides to the growing DNA backbone, matching A to T and C to G, thus creating two identical double-stranded DNA molecules. The two newly created circular chromosomes attach to the inside of the plasma membrane, each at a different spot. The original cell, called the parent cell, then pinches in somewhere between these two spots until it divides into two new cells that are called daughter cells. Each of the daughter cells has an identical two-stranded copy of the original two-stranded circular chromosome. In some procs, such as E. coli bacteria that live in our digestive system, the complete process of binary fission can occur very quickly, often in just 20 minutes. Binary fission is considered a form of asexual reproduction because the daughter cells inherit their DNA from a single parent cell and thus are genetically identical to the parent. And that brings us to our take-home message for section 8.2. In most bacteria and archaea, the genetic information is carried in a single circular chromosome, a closed loop of DNA that is attached at one site to the cell membrane. Generally speaking, eukaryotes have much more DNA than do bacteria and organize it into linear chromosomes within the nucleus. Moving on. Eukaryotic cells don't do every job at once. Instead, they go through phases. They may, for example, spend long periods occupied with activities relating solely to their growth, and then may suspend those activities as they seg into a period devoted exclusively to reproducing themselves. This alteration of activities is called the cell cycle. Before we go further, we need to note an important distinction between the types of cells in the body. All the cells of multicellular eukaryotic organisms can be divided into two types: somatic cells, which are cells forming the body of the organism, and reproductive cells, or germ cells, which are the sex cells, that is, the gametes, the sperm and the egg, and the cells that give rise to them. Somatic cells and reproductive cells use different methods of producing new cells. We will focus first on cell division as it occurs in somatic cells. Then later in the chapter, we will examine cell division that leads to the production of the sex cells. So the cell cycle describes the series of phases in somatic cell division. The two main phases in the cell cycle are interphase, during which the cell grows and prepares to divide, and the mitotic phase, or the M phase, during which cell division occurs. And this cartoon is on page 206 of your book, figure 8.5, and it details the cell cycle for mitosis. So, interphase is further divided into 3 distinct subphases. First is gap 1. During this period, a cell may grow and develop as well as perform its various cellular functions, making proteins, getting rid of waste, and so on. Most cells inhabit the gap 1 phase the majority of the time. However, some cells enter a state called gap 0, which is a resting phase outside the cell cycle in which no cell division occurs. The gap 0 phase may last for days or even years or permanently, as in the case of most neurons and heart muscle cells before resuming cell division. DNA synthesis, or the S phase, is the phase in which the cell begins to prepare for cell division. First, it creates an exact duplicate of each chromosome by replication. Before replication, each chromosome's DNA consists of a single long linear molecule. After replication, each chromosome's DNA has become a pair of identical long linear molecules held together near the center. Then in gap 2, significant growth occurs, along with high rates of protein synthesis in preparation for the division of the cell. Gap 2 differs from gap 1 in its shorter duration and its genetic material now existing in duplicate. The period of the cell cycle that begins with mitosis is called the mitotic phase, or the M phase, and this is a process in which the parent cell's nucleus, including its chromosomes, divide. Mitosis is generally followed by cytokinesis, which may begin before the end of mitosis, during which the cytoplasm divides to form 2 daughter cells. Each daughter cell has a complete set of the parent cell's DNA and other cellular structures. The mitotic phase is usually the shortest period in the eukaryotic cell cycle. There is great variation in how cell types move through the phases of the cell cycle. Animal embryos at one end of a continuum may move through the cell cycle so quickly that they spend almost no time at all in the G1 and the G2 phases. Heart muscle cells and brain neurons at the other end of the continuum may never pass through the cell division phase, remaining in a non-dividing state for decades. Moving on, rates of cell division are regulated by a cell cycle control system, which is a group of molecules, mostly proteins, within a cell that coordinates the events of the cell cycle. This control system functions through a system of checkpoints, critical points in the cell cycle at which progress is blocked and cells are prevented from dividing until specific signals trigger continuation of the process. The signals that trigger the cell cycle transitions commonly consist of growth factors, which provide feedback about the cell's environment and can signal that division is appropriate. Checkpoints in the cell cycle make it possible for cells to 1. Reduce the likelihood of completing cell division when errors have occurred in the process, and 2. Respond to feedback conveying information about the cell's internal and external environments. 3 primary checkpoints regulate the cell cycle in eukaryotes. The G1S checkpoint assesses DNA damage and cell growth. Occurring near the end of the gap 1 phase, this is the point when a cell decides whether or not it will proceed to the S phase and complete cell division, or delay cell division, entering into an extended resting phase, or gap 0. Gap 0 is more precisely defined as a non-dividing, non-growing phase that may include great metabolic activity critical to an organism's proper functioning. As noted earlier, many neurons and muscle cells never leave gap 0, living and functioning for many decades without dividing. Several situations can prevent a cell from passing this G1S restriction point. For example, cells cultured in a laboratory will delay division at this point if the growth medium lacks sufficient nutrients. And if a cell's DNA has been damaged, the cell may be blocked from entering the S phase, and DNA repair mechanisms may be triggered. A malfunction in the G1S checkpoint can allow cells with damaged DNA to continue their division, thereby leading to uncontrolled cell division and cancer. And that brings us to the G2M checkpoint, which assesses DNA synthesis. Just before beginning mitosis, a cell reaches the G2M checkpoint. This checkpoint serves as a mitosis readiness assessment. If the checkpoint is passed, indicating that no DNA damage is detected, the cell initiates the complex process of mitosis. If not, the cell typically undergoes repair of its damaged DNA. Then we have the spindle assembly checkpoint, which assesses anaphase readiness during mitosis. At this point in mitosis, the cell cycle control mechanisms assess whether the chromosomes have aligned properly at the metaphase plate and whether there is appropriate tension on them. If this checkpoint is passed, the cell completes cell division. And that brings us to our take-home message for section 8.3. Eukaryotic somatic cells alternate in a cycle between the mitotic phase, in which cells are divided, and interphase, in which cell growth and other metabolic activities occur during the two gap phases. At a series of checkpoints in the cell cycle, progress is blocked, and cells are prevented from dividing until specific signals trigger the continuation of the process. Moving on. In one of the great understatements in the scientific literature, in their paper describing the structure of DNA, James Watson and Francis Crick wrote, "It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. The researcher's ability to explain how DNA copied itself was a critical feature of the description of DNA structure. After all, every single time any cell in any organism's body divides, the cell's DNA must first duplicate itself so that each of the two new daughter cells has all of the genetic material of the original parent cell. The process of DNA duplication, as we've seen, is called replication." Moving on. But what is it about DNA structure that makes duplication so straightforward? Watson and Crick were referring to the feature of DNA called complementary, meaning that in the double-stranded DNA molecule, the base on one strand always has the same pairing partner called the complementary base on the other strand. In other words, A pairs with T and vice versa, and G pairs with C and vice versa. We've talked about this in a previous chapter. With this consistent pattern of pairing, one strand carries all the information needed to construct its complementary strand. Just before the cells divide, the DNA molecule unwinds and unzips, and each half of the unzipped molecule serves as a template on which the missing half is reconstructed. At the end of the process of reconstructing the missing halves, there are two DNA molecules, each identical to the original DNA molecule, one for each of the two new cells. And then recall from Chapter 6 that the individual units that make up DNA are nucleotides, which have three components: a nitrogenous molecule called a base, a phosphate group, and a molecule of a 5-carbon sugar. Each of the 5 carbon atoms in the sugar molecules is given a number. The nitrogenous base is attached to the 1' carbon, and a hydroxyl group is attached to the 3' carbon, and the phosphate group is attached to the sugar's 5' carbon atom. Moving on. The process of DNA replication occurs in 2 steps. First, unwinding and separation of the 2 strands, and second, reconstruction and elongation of the new complementary strands. A feature of DNA that has important consequences for the process of replication is that its two strands run in opposite directions. The backbone of each strand of a DNA molecule is a long alternation of sugar phosphate, sugar phosphate, sugar phosphate, and so on, going from one end to the other in a 5' to 3' pattern in one strand, and a 3' to 5' pattern in the opposite strand. So first, let's talk about unwinding and separation. Replication begins at a specific site called the origin of replication, where the coiled, double-stranded DNA molecule unwinds and separates into two strands. In prokaryotes, there is a single origin of replication, whereas eukaryotes have multiple origin sites on each chromosome. At the origin of replication, a complex of proteins binds to the DNA. One of the proteins, an enzyme called DNA helicase, unwinds the coiled DNA and separates the two complementary strands. The unwinding and separating of the two DNA strands creates what is called a replication fork. Second is reconstruction and elongation. At the replication fork, a group of several proteins called a replication complex binds to each of the exposed strands. This complex includes the enzyme DNA polymerase, which adds DNA nucleotides with bases that are complementary to the bases on each exposed strand. Replication proceeds in both directions at once from each origin of replication. DNA replication occurs in all types of cells, somatic and reproductive, before cell division. The result of replication is two double-stranded DNA molecules that carry virtually the same genetic information. This makes it possible for the somatic cells of the body that are produced by cell division to have virtually identical genetic composition. Note, though, that the daughter DNA molecules are not completely identical. Replication occurs with a very low rate of errors, about 1 error per several billion base pairs in eukaryotes. Moving on, a variety of mutations, such as mismatched bases and added or deleted segments, can occur during replication or when DNA is damaged by some external source, such as X-rays. Because DNA polymerases perform proofreading functions as well as excision and repair functions, many of these errors are caught and repaired during or after replication or whenever they occur. Not all errors are corrected. And if an error remains, the sequences in a replicated DNA molecule, including the genes, can be different from those in the parent molecule. A changed sequence may ultimately lead to the production of a different protein. When an error is introduced into the DNA of gamete-producing cells, in other words, the cells that divide to produce sperm and eggs, a new gene can sometimes enter a population in the next generation and be acted on by evolution. We will explore this concept in a subsequent chapter. And that brings us to our take-home message for section 8.4. Every time it divides, a cell must first duplicate its DNA so that each of the two new daughter cells has all the genetic material of the original parent cell. The process of DNA duplication, called replication, occurs in 2 steps: unwinding and separation of the 2 strands, then reconstruction and elongation of the new complementary strands. The end result is 2 identical double-stranded DNA molecules. Moving on, look around your room. Dust is everywhere. What is dust? It is primarily dead skin cells. In fact, you and your friends and your family slough off millions of dead skin cells each day, yet your skin doesn't disappear. Why not? Because your body replaces the sloughed off cells through a process of mitosis. Mitosis has just one purpose: to enable existing cells to generate new genetically identical cells. Mitosis is required for growth and replacement. Moving on, Every day, a huge number of cells in an organism must be replaced by mitosis. In humans, this number is in the billions. Nearly all somatic cells of the body, that is, all cells except the sperm and egg-producing cells, undergo mitosis. There are a few notable exceptions, as we've already seen. Heart muscle cells and most neurons, in particular, do not seem to divide, or if they do divide, they do it very slowly. The rate at which mitosis occurs in animals varies dramatically for different types of cells. The most rapid cell division takes place in the bone marrow as red blood cells are produced, as well as in the lining of various tissues and organs. The average red blood cell, for example, circulates for 2 to 4 months and then must be replaced. The cells lining the intestines are replaced about every 3 weeks. Some cells die in a planned process of cellular suicide called apoptosis. This seemingly counterproductive strategy takes place in cells that are likely to accumulate significant genetic damage over time and are therefore at high risk of becoming cancer cells. This process will be described later in this chapter. Cells targeted for apoptosis include many of the cells lining the digestive tract and cells in the liver, two locations where cells are almost constantly in contact with harmful substances. Moving on, prior to mitosis, the parent cell replicates its DNA, creating a duplicate copy of each chromosome. Once this task is completed, mitosis can proceed. During this process, newly duplicated chromosomes are separated into identical sets in 2 separate nuclei. After mitosis, the cytoplasm and the rest of the cell are divided into 2 cells that pinch apart. Where once there was one parent cell, now there are 2 identical daughter cells. Throughout all of the cell cycle except mitosis, chromosomes are uncoiled and spread out in a diffuse way like a mass of spaghetti. Because they are so stretched out, they are not dense enough to be visible under a light microscope. However, just before mitosis begins, 2 important events occur. First, the chromosomes replicate, beginning 2 identical linear DNA molecules that are held together at the centromere. Throughout mitosis, until the centromeres separate, each of the identical DNA molecules is called a chromatid. Together, the 2 are called sister chromatids. In the second event preceding mitosis, these sister chromatids begin the process of condensation, in which they coil tightly and become compact, in contrast to the uncondensed and tangled state of the chromosome prior to replication during most of interphase. The genetic material appears as X-shaped chromosomes in most photos because once it has condensed in preparation for cell division, it is coiled tightly. Only then is it thick enough to be seen and photographed under a light microscope. Thus, the X-shaped image is not simply a chromosome, but a replicated chromosome. Because the sister chromatids need space to separate, the membrane around the nucleus is dismantled and disappears early in mitosis. At the same time, the spindle is assembled. The spindle consists mostly of hollow tubules of proteins called microtubules, which are part of the cell's cytoskeleton. They resemble a group of threads stretching across the cell between its 2 ends or poles. In animal cells, the threads originate and spread out at each pole from structures called centromeres, which contain a pair of centrioles and a mass of proteins that anchor the microtubules. These threads, known as spindle fibers, attach to the centromeres and pull the sister chromatids to the middle of the cell. During mitosis, they'll eventually pull the chromatids apart as cell division proceeds. And that brings us to our take-home message for section 8.5. Mitosis is the process by which cells duplicate themselves. Mitosis follows chromosome replication and leads to the production of two genetically identical daughter cells from one parent cell. This makes it possible for organisms to grow and to replace cells that die. So, let's look at the process of mitosis in a bit more detail, keeping in mind that the ultimate result is the production of two cells with identical sets of chromosomes. Moving on. Processes essential to cell division take place even before the mitotic phase of the cell cycle begins. During the DNA synthesis part of interphase, sister chromatids are formed as every chromosome replicates itself. Each pair of sister chromatids is held together at the centromere. The actual process of cell division occurs in 4 stages. First is prophase. Prophase begins when sister chromatids condense. At this point, the spindle forms and the nuclear envelope breaks down. Second is metaphase. After condensing, the pairs of sister chromatids line up at the cell's center, pulled by spindle fibers that are attached to a disc-like group of proteins that develops on the centromeres. By the end of metaphase, all the chromatid pairs are lined up in an orderly fashion, straddling the center in a single file congregation called the metaphase plate. It should be noted that the chromatids are at their most condensed during this part of mitosis. Moving on, this brings us to anaphase, and in anaphase, the spindle microtubules attached to the centromeres begin pulling each of the chromatids in a sister chromatid pair towards opposite poles of the cell. Each pair of sister chromatids, the centromeres, separate as one DNA molecule is pulled in one direction and the other identical DNA molecule is pulled in the opposite direction. At the end of anaphase, one full set of chromosomes is at one end of the cell and another identical full set is at the other end. And then in telophase, 2 full identical sets of chromosomes collected at either end of the cell, the parent cell, is prepared to divide into 2 genetically identical cells. In this last stage, called telophase, the chromosomes begin to uncoil, fading from view. The nuclear membranes reassemble. The process of mitosis is generally accomplished by cytokinesis, which typically begins during telophase. During cytokinesis, the cell's cytoplasm is divided into approximately equal parts and the cell divides, with some of the organelles going to each of the 2 new cells. When cytokinesis is complete, the 2 new daughter cells enter interphase and begin the business of being cells. Usually, mitosis occurs without errors, and in situations in which new or replacement cells are needed. In rare cases, however, something goes wrong, and that can cause cell division to proceed unchecked. In such cases, cancer can arise. That brings us to our take-home message for section 8.6. The ultimate result of mitosis and cytokinesis is the production of 2 genetically identical cells. Moving on, cell growth and division are necessary to the health of most organisms, but too much of a good thing can be bad. Cancer is characterized by unrestrained cell growth and division that can damage adjacent tissues. Cancer-causing mutations seem to affect 2 different types of genes, those that stimulate cell growth and those that restrain it. Cancer can cause serious health problems and is the second leading cause of death in the United States after heart disease. Moving on, cancer occurs when disruption of the DNA in a normal cell interferes with the cell's ability to regulate cell division. DNA disruption can be caused by chemicals that trigger mutation in the DNA or by sources of high energy such as X-rays, the sun, or nuclear radiation. Cancer can even be caused by some viruses. Whatever the cause, once a cell loses control over its cell cycle, cell division can proceed unrestrained. Cancer cells have several features that distinguish them from normal cells. First, cancer cells lose their contact inhibition. Most normal cells divide until they touch other cells or collections of cells or tissues. At that point, they stop dividing. Cancer cells, however, ignore the signal that they are at high density and continue to divide. Second, cancer cells can divide indefinitely. As we saw in a previous section, most normal human cells can divide 80 to 90 times. After that point, a cell may continue living but loses the ability to divide. In contrast, cancer cells never lose their ability to divide and continue to do so indefinitely, even in the presence of conditions that would normally halt the cell cycle before cell division. Third, cancer cells have reduced stickiness. Cells are normally held together by adhesion molecules, proteins within the cell membranes. And cancer cells, too, usually group together forming a tumor. But the membranes of cancer cells tend to have reduced adhesiveness, causing them to stick to each other less than do non-cancerous cells. Moving on, tumors caused by excessive cell growth and division are of two very different types, benign and malignant. Benign tumors, such as many moles, are just masses of normal cells that do not spread. Most do not need to be removed. Those that do can usually be removed safely without any lasting consequences. Malignant tumors, however, are the result of unrestrained growth of cancerous cells. Malignant tumors shed and spread cancer cells. This is a process called metastasis. In this process, cancer cells separate from a tumor and invade the circulatory system and lymphatic pathways, then spread to different parts of the body where they can cause the growth of additional tumors. How does cancer actually kill the organism? Somewhat surprisingly, it's not because of some toxic action of the cancer cells themselves. Like an invasive weed in a garden sucking the nutrients away from vegetables and edging out for space wanted plants, a tumor uses up nutrients and energy, takes up more and more space, and presses against neighboring cells and tissues. Eventually, the tumor may block other cells and tissues from carrying out their normal functions and even kill them. This cell dysfunction or cell death can have disastrous consequences when the job of the affected normal tissue is to control critical processes such as breathing, heart function, or the detoxification processes of the liver. Moving on. To treat cancer, the rapidly dividing cells must be removed surgically or killed, or at least their division must be slowed down. Currently, the killing and slowing down are done in two ways. One is chemotherapy and the other is radiation. In chemotherapy, drugs that interfere with cell division are administered with the goal of slowing down the growth of tumors. Because these drugs interfere with rapidly dividing cells throughout the body, not just the rapidly dividing cancer cells, they can have very unpleasant side effects. In particular, chemotherapy drugs disrupt normal systems that rely on the rapid and constant production of new cells. For instance, chemotherapy often causes extreme fatigue and shortness of breath because it reduces the rate at which blood cells are produced, thus limiting the amount of oxygen that can be transported throughout the body. By interfering with the division of bone marrow stem cells, chemotherapy also reduces the production of platelets and white blood cells and thus increases bruising and bleeding, as well as a susceptibility to infection. The rapidly dividing cells within the hair follicles are also affected by chemotherapy and radiation. As a consequence, many people lose their hair when undergoing treatments. It usually grows back, however, when the treatments stop. Like chemotherapy, radiation works by disrupting cell division. However, radiation therapy is more targeted than the drugs used in chemotherapy. This treatment directs high-energy radiation at the part of the body where the tumor is located. As with chemotherapy, radiation is not perfect, and nearby tissue is often harmed. Although there is no complete cure for cancer, many potentially successful therapies for treatment and preventions are on the horizon. Extensive research is being conducted on the mechanisms by which genes controlling the cell cycle are damaged in cancer cells, for example, and how such damage might be prevented or reversed. And that brings us to our take-home message for section 8.7. Cancer is unrestrained cell growth and cell division, which can lead to large masses of cells that may cause serious health problems. It often results from mutations in genes important in controlling the cell cycle. Moving on, there are two ways in which an organism can reproduce. Many organisms, including bacteria, fungi, and some plants and animals, undergo asexual reproduction, in which a single parent produces identical offspring. Other organisms, including most animals and plants, undergo sexual reproduction, in which offspring are produced by the fusion of two reproductive cells in the process of fertilization. And some species, particularly among plants, can use both methods. In sexual reproduction, because a combination of DNA from two separate individuals is passed on to offspring, the resulting offspring are genetically different from their parents and, for reasons explained later, from one another. But what would happen if sexually reproducing organisms, humans included, produced reproductive cells through mitosis? Then, both parents would contribute a full set of genes, that is, 23 pairs of chromosomes in humans, to create a new individual. And this new individual, this new offspring, would therefore inherit 46 pairs of chromosomes in all. And when that individual reproduced, if he or she contributed 46 pairs of chromosomes, and his or her mate also contributed 46 pairs, their offspring would have 92 pairs of chromosomes. Where would it end? The genome would double in size with every generation. In sexually reproducing organisms, the solution to chromosome overload is meiosis. This process enables organisms to make gametes, special reproductive cells that have half as many chromosomes as the rest of the cells in the organism's body, the somatic cells. In humans, for example, each gamete cell has only one set of 23 chromosomes rather than two sets. Moving on, in genetics, the term diploid refers to cells that have two copies of each chromosome. In humans, two sets of 23 chromosomes for 46 chromosomes in all. And the term haploid refers to cells that have one copy of each chromosome. Thus, somatic cells are diploid, and gametes, the cells produced in meiosis, are haploid. During fertilization, two haploid cells, each with one set of 23 chromosomes, merge and create a new individual with the proper diploid genome of 46 chromosomes in humans. This new individual, through meiosis, will also produce haploid gametes that have only a single set of 23 chromosomes. With sexual reproduction, then, diploid organisms produce haploid gametes that fuse at fertilization to restore the diploid state. In this way, meiosis maintains a stable genome size in a species. Moving on, although some variations are seen in this pattern of alternation between haploid and diploid states, in most cases, multicellular animals produce haploid cells for reproduction. When two haploid gametes come together to form a diploid fertilized egg, multiple cell divisions by mitosis then produce a diploid multicellular animal. Meiosis achieves more than just a reduction in the amount of genetic material in the gametes. As a diploid individual, you have two copies or two alleles of every gene in every somatic cell. one from your mother and one from your father. As a consequence of meiosis, gametes have one allele for each trait rather than two. Which of these two alleles is included in each gamete cannot be predicted. Each egg or sperm carries a unique set of alleles produced with a varied combination of material and parental alleles. All offspring resemble their parents, yet none of them exactly the same way. In all, meiosis has two important outcomes. It reduces the amount of genetic material in the gametes, and it produces gametes that differ from one another with respect to the combinations of alleles that they carry. And that brings us to our take-home message for section 8.8. Meiosis is the process by which reproductive cells are produced in sexually reproducing organisms. This results in gametes that have only half as much genetic material as the parent cell and that differ from one another in the combinations of alleles that they carry. Moving on, mitosis is an all-purpose process for cell division. It occurs all through the body, all of the time. Meiosis, on the other hand, takes place in just a single place, the gonads, the ovaries and testes. And it takes place for just a single reason, the production of gametes, that is, the sperm and the eggs. Meiosis starts with a specialized diploid cell found in the gonads that is capable of undergoing meiosis. Thus, for humans, meiosis starts with a cell that has 46 chromosomes. These include a maternal copy and a paternal copy of each of the 22 chromosomes. Each of these pairs is called a homologous pair or homologs, along with two additional chromosomes, one from each parent, called the sex chromosomes. Moving on, before meiosis can occur, and just as we saw with mitosis, each of these 46 chromosomes is duplicated during the cell's interphase. This means that when meiosis begins, after replication, each cell has 92 strands of DNA, two sister chromatids for each of the 46 chromosomes. Whereas mitosis includes only one cell division, cells undergoing meiosis divide twice. In the first division, the homologs separate. In other words, for each of the 23 pairs of chromosomes, the maternal sister chromatid pairs and the parental sister chromatid pairs separate randomly into new cells. In the second division, each of the two new cells divides again, so that each of the four daughter cells contains a single chromosome from the homologous pair. At the end of meiosis, there are four new cells, each of which has 23 strands of DNA, that is, 23 chromosomes in animals. None of these four cells will undergo any further cell division. They do not become parent cells for a new cycle of cellular division. Before meiosis begins, every chromosome creates an exact duplicate of itself by replication. The chromosomes that were each a single long linear piece of genetic material become a pair of identical long linear pieces held together at the centromeres. Meiosis begins following replication and proceeds through 8 steps. And that brings us to our next slide that details meiosis 1. The first meiotic division takes place in 4 stages. Prophase 1, where the chromosomes condense and cross over. This is by far the most complex of all the phases of meiosis. As in mitosis, prophase 1 begins with all of the replicated genetic material condensing. So as the sister chromatids become shorter and thicker, the homologous chromosomes come together. This is where the process diverges from mitosis. The homologous chromosomes, each of which has become a pair of identical chromatids, line up, touching. Under a microscope, the two homologous pairs of sister chromatids appear as pairs of X's lying on top of one another. At this point, the sister chromatids that are next to each other do something that make every sperm and egg cell genetically unique. They swap little segments of DNA. Some of the genes that you inherited from your mother may get swapped onto the strand of DNA you inherited from your father, and vice versa. This possible outcome of the crossing over of portions of the chromatids is called genetic recombination, or more commonly, we just call it recombination. It can take place at several places, up to dozens on each chromatid. As a result of recombination, every sister chromatid possesses a unique mixture of your genetic material. Note that crossing over takes place only during the production of gametes in meiosis. It does not occur during mitosis. Following crossing over, the nuclear membrane then disintegrates. We'll talk more about crossing over later. So metaphase 1 is where all chromosomes line up along the center of the cell. So after crossing over and after disintegration of the nuclear envelope, each pair of homologous chromosomes moves to the center of the cell, pulled by the spindle fibers to form the arrangements called the metaphase plate. Keep in mind that each pair of homologous chromosomes includes the maternal and paternal versions of the chromosome with crossed over segments, and the replicated copy of each, making 4 strands in all. The maternal and paternal sister chromatid pairs line up at the metaphase plate in a random fashion. This is called random assortment, so that the pairs of sister chromatids pulled towards each pole are a mix of maternal and paternal sister chromatids. As a result of random assortment, all the products of meiosis are genetically unique. Imagine all the different combinations that can occur in a species with 23 pairs of chromosomes. For chromosome pair 1, perhaps the maternal homolog will go to the top pole and the paternal homolog will go to the bottom pole. And for pair 2, perhaps the maternal homolog will also go to the top and the paternal homolog will go to the bottom. For each of the 23 pairs, you cannot predict which will go to the top and which will go to the bottom. There is a huge number of possible combinations. In anaphase 1, this phase is the beginning of the first cell division that occurs during meiosis. In this phase of meiosis, the spindle fibers pull the homologs apart toward opposite poles of the cell. One of the homologs, consisting of 2 sister chromatids, goes to one pole, the other to the opposite pole. And then in telophase 1 and cytokinesis, the nuclear membranes reassemble around the sets of sister chromatid pairs and 2 daughter cells form. After the pairs of chromatids arrive at the 2 poles of the cell, nuclear membranes reform. The cytokinesis stage occurs when the cytoplasm divides and the cell membrane pinches the cell into 2 daughter cells. Each daughter cell has a nucleus that contains the genetic material, 2 sister chromatids for each of the 23 chromosomes. So moving on to meiosis 2, there is a brief interphase after the first division of meiosis. In some organisms, the DNA molecules, now in the form of chromatid pairs, briefly uncoil and fade from view. In others, the second part of meiosis begins immediately. In the brief interphase before prophase 2, no replication of any of the chromosomes occurs. The second part of meiosis, like the first division of meiosis, is a 4-phase process. So in prophase 2, the chromosomes recondense. The second division of meiosis begins with prophase 2. The genetic material in each of the 2 daughter cells once again coils tightly, making the pairs of chromatids visible under the light microscope, and the nuclear membrane again disintegrates. But unlike in prophase 1, no crossing over occurs during prophase 2. Then in metaphase 2, the sister chromatid pairs line up at the center of the cell. In each of the two daughter cells, the sister chromatid pairs, each pair appearing as an X, are pulled to the center of the cell by spindle fibers attached to the centromeres. The congregation of all the genetic material in the center of each daughter cell is visible as a flat metaphase plate. In anaphase 2, the sister chromatids are pulled to the opposite sides of the cell. During this phase, the fibers attached to the centromeres begin pulling each chromatid in each of the 23 sister chromatid pairs toward opposite poles of the daughter cell. When anaphase 2 is finished, each of what will become the 4 daughter cells, you could think of them as granddaughter cells, I guess at this point, has one copy of each of the 23 chromosomes. And that brings us to telophase 2 and cytokinesis, where the nuclear membranes reassemble and the 2 daughter cells pinch into 4 haploid gametes. After the sister chromatids for all 23 chromosomes have been pulled to opposite poles, the cytoplasm divides. The cell membrane pinches each cell into 2 new daughter cells. Nuclear membranes begin to reform, and the process comes to a close. In humans, the outcome of one diploid cell undergoing meiosis is the creation of 4 haploid daughter cells, each with a set of 23 individual chromosomes. These chromosomes contain a combination of traits from the individual's diploid set of chromosomes. And that brings us to our take-home message for section 8.9. Meiosis in animals occurs only in gamete-producing cells. It is preceded by DNA replication and consists of 2 rounds of cellular division, one in which homologous pairs of sister chromatids separate, and a second in which the sister chromatids separate. The final product of meiosis in a diploid organism is 4 genetically dissimilar haploid gametes. Moving on, for all sexually reproducing organisms, there is just one way to distinguish males from females, and it's nothing visible to the naked eye. When there are 2 sexes, as in the case for nearly every sexually reproducing animal and plant species, the females produce the larger gamete and the males produce the smaller, more motile gamete. Meiosis works differently in males and females. The female gamete is larger than the male gamete because it has more cytoplasm. During the production of sperm, meiosis takes place just as described in section 8.9, resulting in 4 evenly sized cells that become sperm. However, during egg production, the cell divides in telophase 1, and the genetic material is evenly divided, but nearly all of the cytoplasm goes to one of the cells and almost none goes to the other. The smaller cell, called a polar body, degrades almost immediately in most animals. Then, in the second meiotic division of the larger cell, there is again an unequal division of cytoplasm. As in the first division, one of the new cells gets nearly all of the cytoplasm, and the other gets almost none, forming another polar body. The net result of meiosis in the production of eggs is one large egg with lots of cytoplasm and 2 or 3 small polar bodies with very little cytoplasm that degrade and will never function as gametes. Ultimately, whether egg or sperm, each gamete ends up with just one copy of each chromosome. That way, the fertilized egg that results from the fusion of sperm and egg carries 2 complete sets of chromosomes, so the developing individual will be diploid. The egg's extra cytoplasm contains a large supply of nutrients and other resources to help with the initial development of the organism following fertilization. I'd also like to point out here, at least in humans, that the female never completes meiosis unless she is fertilized. And that brings us to our take-home message for section 8.10. In species with two sexes, including nearly every sexually reproducing plant and animal species, females are the sex that produces the larger gamete, and males produce the smaller gamete. Each gamete ends up with just one copy of each chromosome. Moving on, genetically speaking, there are two ways to create unique individuals. The obvious way is for an organism to carry an allele that is not present in any other individuals. Alternatively, and equally successful in creating uniqueness, an individual can carry a collection of alleles that has never before existed in another individual. Both types of novelty introduce important variation into a population of organisms. The process of crossing over, or genetic recombination, which occurs during prophase 1 in meiosis, creates a significant amount of the second type of variation. So let's look at crossing over more carefully. Take, for example, the homologous pair of human chromosome 15. It includes two copies of chromosome 15, one copy from your mother, which you inherited from the egg that was fertilized to create you, and one copy from your father, which you inherited from the sperm that fertilized the egg. Each chromosome in the pair carries the same genes, but because they came from different people, they do not necessarily have the same alleles. Once the sister chromatids of the homologous chromosome pairs line up in prophase 1 so that the four chromatids in two pairs lie very close together, regions that are close together can swap segments. A piece of one of the maternal chromatids, perhaps including the first 100 genes on the strand of DNA, may swap places with the same segment in a paternal chromatid. Elsewhere, a stretch of 20 genes in the middle may be swapped from the other maternal chromatid with one of the paternal chromatids. The points at which chromatids exchange genetic material during recombination are called chiasmata. Every time a swap of DNA segments takes place, an identical amount of genetic material is exchanged, so all four chromatids still contain the complete set of genes that make up the chromosome. The combination of alleles on each chromatid, though, is now different. Suppose genes relating to eye color and height are present on a particular chromosome. After crossing over, a chromatid that carried instructions for brown eyes and short height may now carry instructions for brown eyes and tall height. All of the alleles from your parents are present on one DNA molecule or another, but the combination of alleles and the traits that they determine are linked together on a single chromatid, and this is new. And when a gamete, let's say it's an egg, carrying a new combination of alleles is fertilized by a sperm, the developing individual will carry a completely novel set of alleles. So without creating new versions of any traits, such as yellow eyes or purple hair, crossing over creates gametes with collections of alleles that may never have existed together before. And that brings us to our take-home message for section 8.11. Although it doesn't create new versions of any alleles, crossing over during the first prophase of meiosis creates gametes with combinations of alleles and of traits they determine that may never have existed before. This variation is important for evolution. Moving on. As we've seen, cells and organisms can reproduce through asexual or sexual reproduction. Is one method better than the other? Well, it depends. A better question to ask is, what are the advantages and disadvantages of each method, and under what conditions do the benefits outweigh the costs? So what are the advantages of sexual reproduction? Sexual reproduction leads to offspring that are genetically different from one another and from each parent through three different processes. One is the combining of alleles from two parents at fertilization. With sexual reproduction, a new individual comes from the fusion of gametes from two different individuals. Each of these parents comes with his or her own unique set of genetic material. Second, crossing over during the production of gametes. Crossing over during prophase 1 of meiosis causes every chromosome in a gamete to carry a mixture of an individual's maternal and paternal genetic material. And third is the shuffling and reassortment of homologs during meiosis. When homologs for each chromosome are pulled to the opposite poles of the cell during the first division of meiosis, that is anaphase 1, the maternal and paternal homologs are randomly separated. As we saw in section 8.9, a staggeringly large number of different combinations of maternal and paternal homologs could end up in each gamete. The variability among the offspring produced by sexual reproduction enables populations of organisms to cope better with changes in their environment. After all, if the environment is gradually changing from one generation to the next, the production of many genetically different offspring increases the likelihood that some of the offspring will carry sets of genes particularly well suited to the new environment. Over time, populations of sexually reproducing organisms can quickly adapt to changing environments. It's like buying a different lottery ticket for a drawing. The more tickets you buy, the more likely it is that one of them will be a winner. But what are the disadvantages of sexual reproduction? Well, in the yellow dung fly, males sometimes wrestle each other for mating access to a female. The female awaits the outcome of the battle in a pile of dung. Occasionally, females will drown in the dung pile as they wait. Males, too, can be at risk during reproduction. This is seen most dramatically in species such as praying mantises and black widow spiders, in which the female will attempt to eat the male and often succeed in doing so during or after mating, gaining not just the gametes but a meal. Dangers associated with mating are one of the downsides of sexual reproduction. As we see with the dung flies, sex can be a risky proposition because organisms make themselves vulnerable to predation, disease, and other calamities during mating and reproduction. There are other risks too. First, when an individual reproduces, only half of its offspring's alleles will come from that organism. The other half will come from the other parent. The complex cellular division required for sexual reproduction offers opportunities for mistakes, sometimes leading to chromosomal disorders. With asexual reproduction, an individual produces nearly identical offspring, so there is very efficient transfer of genetic information from one generation to the next. Second, with sexual reproduction, it takes time and energy to find a partner. This is energy that asexual organisms can devote to additional reproduction. So what are the advantages of asexual reproduction? Because asexual reproduction involves only a single individual, it can be fast and easy. Some bacteria can divide, forming a new generation every 20 minutes. Likewise, for organisms in isolated habitats or those establishing new populations, asexual reproduction can be advantageous. Asexual reproduction is efficient too. Offspring carry all of the genes that their parent carried, so they are genetically identical. If the environment is stable, it is beneficial for organisms to produce offspring as similar to themselves as possible. But what are the disadvantages of asexual reproduction? The downside to asexual reproduction is that the more closely an offspring's genome resembles that of its parent, the less likely it is that said offspring will be suited to the environment when it changes. Conversely, by producing offspring that are more genetically varied from one another, parents are more likely to successfully leave offspring. In the end, we still see large numbers of species that reproduce asexually and large numbers that reproduce sexually. It seems that conditions favoring one or the other occur in the great diversity of habitats in our world. That we see both asexual and sexual reproduction also highlights the recurring theme in biology that there often is more than one way to solve a problem. And that brings us to our take-home message for section 8.12. Cells and organisms can reproduce by mitosis and asexual reproduction, and by meiosis and sexual reproduction. Asexual reproduction can be efficient but leads to offspring that are genetically identical to their parents, which could be a disadvantage if the environment is changing. Sexual reproduction is less efficient but yields offspring that are genetically different from one another and from either parent. Moving on, in humans, the sex of a baby is determined by its father. The sequence of events involved in sex determination is instigated by one special pair of chromosomes, the sex chromosomes, which carry the information that directs a growing embryo to develop as a male or as a female. Chromosomes can be divided into two different types: one pair of sex chromosomes and 22 pairs of non-sex chromosomes. The human sex chromosomes are called the X and Y chromosomes. And you can see a picture of those on your slide. This is figure 8.29 in your book on page 226. And you can see the Y chromosome in the blue, how small it is. It is the smallest chromosome in the human body. So how do the X and Y chromosomes differ from other chromosomes? All the genetic information is stored on the chromosomes in all of the cells of an organism's body, but most of this information is not sex-specific. That is, if you are building an eye or a neuron or a skin cell or a digestive enzyme, it doesn't matter whether it is for male or for a female. The instructions are the same for both sexes. Some genetic information, however, instructs the body to develop into one sex or the other. That information is found on the sex chromosomes. Moving on, an individual has two copies of all the non-sex chromosomes, and they are also called autosomes. One copy is inherited from the mother, one copy is inherited from the father. Individuals also have two copies of the sex chromosomes. Males have one copy of the X chromosome and one copy of the Y chromosome, and females have two copies of the X chromosome. So how does the father determine the sex of the baby? Well, during meiosis in females, the gametes that are produced carry only one copy of each chromosome, including the sex chromosomes. Half of the gametes receive a copy of one of the X chromosomes, and half receive a copy of the other X chromosome. Thus, every egg has an X for its one sex chromosome. However, during meiosis in males, the sperm that are produced also carry one copy of each chromosome, including the sex chromosomes. In this case, however, half of the sperm inherit the X chromosome and the other half inherit the Y chromosome. During fertilization, an egg bearing a single X chromosome is fertilized by a sperm bearing either an X chromosome or a Y chromosome. When the sperm carries an X, the baby will have two X chromosomes and will develop as a biological female. When the sperm carries a Y, the baby will have an X and a Y, and so will develop as a biological male. Physically speaking, the X and Y chromosomes look very different from each other. The X chromosome is relatively large and carries a great deal of genetic information relating to a large number of non-sex-related traits. The Y chromosome, on the other hand, is tiny and carries genetic information about only a very small number of traits. The genetic instructions on the Y chromosome set off a cascade of gene activity instructing the fetal gonads to develop as testes rather than ovaries. In the absence of instructions from a Y chromosome, the fetus develops as a female. Once this is done, very little additional genetic input from the Y chromosome is necessary. Instead, the hormones produced by the testes, or, in the absence of the Y chromosome, by the ovaries, generally direct the rest of the body to develop as a male or female. Moving on. A variety of other methods of sex determination are used in other species. In most plants, There aren't even distinct male and female individuals. Every individual produces male and female gametes in separate reproductive parts on the same plant. The same goes for all earthworms and garden snails. Such organisms we can call bisexual, or another word for them is hermaphrodite, because a single individual produces both male and female gametes. Among birds, the female gametes have one copy of either of the two types of sex chromosome, whereas the male gametes all have a copy of one type. Thus, the sex of bird offspring is determined by the female. In ants, bees, and wasps, sex is determined by the number of chromosome sets that an individual possesses. Specifically, males are haploid and females are diploid. A female can fertilize an egg with sperm she has stored after mating, producing a female. Alternatively, she can lay an unfertilized egg, which will develop into a male. And in some species, sex determination is controlled by the environment. In most turtles, for example, the offspring's sex is determined by the temperature at which the eggs are kept. And that brings us to our take-home message for section 8.13. A variety of methods are used for sex determination across the world of plant and animal species. In humans, the sex chromosomes carry information that directs a growing fetus to develop as either a male or a female. Moving on, reproduction becomes riskier for women as they become older. Increasingly, their gametes contain incorrect numbers of chromosomes or chromosomes that have been damaged. These problems can lead to adverse effects on the offspring that range from minor to fatal. Parents can request a quick test for some common genetic problems even before their baby is born. This information is available from an analysis of an individual's karyotype, which is a visual display of the complete set of chromosomes. A karyotype can be developed for adults or children, but it is most commonly done for a fetus. A karyotype is a useful diagnostic tool because it can be prepared very early in the fetus's development to assess whether it has an abnormality in the number of chromosomes or in their structure. Because the test shows all of the chromosomes, even the sex chromosomes, it also reveals the sex of the fetus. Preparing a karyotype takes 5 steps. First, cells are obtained from the individual. Second, the cells are cultured in a test tube with nutrients so that they divide. After a period of cell division, the cells are treated with a chemical that stops them exactly midway through cell division at a time when the chromosomes are coiled thickly and more visible than normal. And fourth, the cells are placed on a microscope slide and a stain is added that binds to the chromosomes, making them even more visible. Finally, the chromosomes are arranged by size and shape and displayed on a monitor where they can be assessed. Moving on, collecting cells from a fetus is more challenging than taking a small blood sample, as is typical for testing adults and children. 2 different methods can be used for collecting fetal cells. Amniocentesis, in which a needle is inserted into the amniotic sac and fluid is removed, and chronic villus sampling, in which tissue is removed from the placenta. Moving on, the resulting karyotype, whether from amniocentesis or CVS, will reveal whether the fetus carries extra copies or lacks copies of any chromosomes. Of all the chromosomal disorders detected by karyotyping, Down syndrome is the most commonly observed. Named after John Langdon Down, the doctor who first described it in 1866, the syndrome is revealed by the presence of an extra copy of chromosome 21. For this reason, the condition that causes Down syndrome is also called trisomy 21. Affecting about 1 in every 1,000 children born, Down syndrome is characterized by a suite of physical and intellectual characteristics that includes learning disabilities, a flat facial profile, heart defects, and increased susceptibility to respiratory difficulties. Moving on, Down syndrome and other disorders caused by missing chromosomes or an extra copy of a chromosome are a consequence of non-disjunction. That is the unequal distribution of chromosomes during cell division. Non-disjunction can occur at two different points during meiosis: when homologs fail to separate during meiosis 1, or when sister chromatids fail to separate during meiosis 2. In both cases, non-disjunction results in an egg or sperm with 0 or 2 copies of a chromosome rather than a single copy. Any of the chromosomes can fail to separate during cell division, but the ramifications of trisomy, having an extra copy of one chromosome in every cell, are greater for chromosomes with large numbers of genes. When trisomy occurs for chromosomes with greater numbers of genes, the developing embryo is less likely to survive birth. Consequently, we tend to see cases of trisomy that involve only the chromosomes with the fewest genes, such as chromosomes 13, 15, 18, 21, and 22. In fact, observations show that trisomy 1 is never seen. All such fertilized eggs die before implantation into the uterus. Trisomy 13 occurs 1 in 20,000 newborns, and most die soon after birth, while trisomy 21, as we've seen, occurs 1 in 1,000 newborns, many of whom live long lives. As we mentioned at the beginning of this section, as women become older, they face increased problems associated with reproduction. Notably, as women age, their gametes tend to have more errors. The reason is that eggs begin meiosis near the time the woman is born, and they do not complete meiosis until they are fertilized. During those decades, the cells may develop some problems, including a reduction in the size and stability of the spindle fibers and reduced cohesion between sister chromatids. That will interfere with normal cell division. In men, the cells that undergo meiosis are relatively young because new sperm-producing cells are produced every couple of weeks after puberty. And that brings us to our take-home message for section 8.15. A karyotype is a visual display of a complete set of chromosomes used as a diagnostic tool to assess whether a fetus shows evidence of an abnormality in chromosomes. Moving on, it is usually fatal to have one too many or one too few of the autosomes. However, many individuals are born lacking one of the sex chromosomes or having one or more additional X or Y chromosomes, and they usually survive. We can get a glimpse into the role of the sex chromosomes by looking at the consequences of having an abnormal number of these chromosomes. In each of the following cases, the condition is caused by non-disjunction of the sex chromosomes during the production of sperm or eggs. Approximately 1 in 5,000 females carries only one X chromosome and no Y chromosome, exhibiting a condition called Turner syndrome, denoted as X or sometimes X0, XO. This is the only condition in humans in which a person can survive without one of a pair of chromosomes. Instead of having 46 chromosomes in every cell, these individuals have only 45. As common as Turner syndrome is, in 98% of the fertilized eggs in which this condition occurs, the embryo is spontaneously aborted long before the fetus can come to term. The lack of a second sex chromosome has both physical and cognitive consequences, including shorter than average height, a web of skin between the neck and shoulders, immature ovaries, underdeveloped secondary sexual characteristics, and some learning difficulties. An individual who has 2 X chromosomes is biologically female An individual with an X and a Y is biologically male. But what happens when both of those conditions exist? An individual who carries two X chromosomes and a Y chromosome, a condition known as Klinefelter syndrome, develops as male. This is because, as we saw earlier, the Y chromosome carries genetic instructions that cause fetal gonads to develop as testes. If these instructions are absent, the fetal gonads develop as ovaries. The extra X chromosome, however, does affect the development of males with Klinefelter syndrome, although these effects can be reduced through treatments such as testosterone supplementation. Approximately 1 in 1,000 males have the genotype XXY, making this one of the most common genetic abnormalities in humans. Klinefelter syndrome has some physical and cognitive consequences, including smaller than average testes and low testosterone, such that men are almost always infertile, reduced facial and chest hair, and some breast development. Taller than average height and language impairment in childhood and throughout life. Some individuals have more than one extra X chromosome with the karyotypes XXXY or even XXXXY. These males also exhibit the symptoms of Klinefelter syndrome, but more frequently have an intellectual disability. A hermaphrodite is an individual with functioning male and female reproductive organs capable of producing both male and female gametes. Sometimes this is called intersex. Often it is mistakenly assumed that a person with Klinefelter syndrome must be a hermaphrodite because he has both two X chromosomes, which would usually make an individual female, and a Y chromosome with an X chromosome, usually making an individual male. Hermaphroditism is common among invertebrates and occurs in some fish and other vertebrates, but contrary to urban legends, human hermaphrodites do not exist. Some men with Klinefelter syndrome may have some features typical of the opposite sex. However, they do not produce female gametes and so are not hermaphrodites. Individuals with sex characteristics that preclude definitive identification as male or female are called intersex. Moving on, there is no official name for the condition in which an individual has one X chromosome and two Y chromosomes. But I've heard it called Swyer syndrome in the past. This chromosomal abnormality occurs in approximately 1 in 1,000 males and in about 1 in 235 males who are 6 feet or taller. There are no distinguishing features at birth to indicate that an individual carries an extra Y chromosome, and the vast majority of affected individuals live their lives without even knowing they have an extra chromosome in every cell. Several consequences of the Swyer's condition have been well documented, including taller than average height, moderate to severe acne, and potentially lower than average intelligence. Although an error during meiosis in either the father or the mother can cause Klinefelter or Turner syndrome, it is only an error in meiosis in a male that can give rise to an XYY child. Why? Because only males carry Y chromosomes. Consequently, an XYY child must have received both of his Y chromosomes from his father. This means that the father's sperm cell must have contained 2 copies of the Y as a result of non-disjunction during meiosis in the father. And individuals with 3 X chromosomes occur at a frequency of about 1 in 1,000 women. Very few studies on this condition have been completed, although initial observations suggest that XXX females are slightly taller than average but have no obvious physical or intellectual problems. In contrast to individuals who have Turner syndrome, most XXX females are fertile. And that brings us to our take-home message for section 8.16. Although it is usually fatal to have one too many or one too few of the non-sex chromosomes, individuals born with only a single sex chromosome that is an X or with an additional X or Y chromosome usually survive, though often with physical and or intellectual problems.